So our next and last speaker for this morning is Guillaume Salbreux from the Crick Institute in London. He'll talk about the physics of epithelial flows and folds.
Okay, thank you very much, Thomas, for organizing this great symposium. It's really a pleasure to hear all these talks. And so I'm at the Francis Crick Institute in London. I'm a physicist, and I'm going to tell you a bit about epithelial flows and folds. And so those are a few examples of developmental processes. So many of you are interested in development, so you probably have seen this before. And in Drosophila and zebrafish, and the common points in these movies is that one can see the collective motion of many cells, which together manage to establish the shape of the organism. And this process involves mechanics, because the cells have to exert forces on each other, and patterning of the tissue. And we want to understand on the long term how these two aspects work together. One common point of the movies I've just shown is that they involve the motion of epithelia, so tissues which have a thickness of a single cell. Inside the epithelium, the cells are connected to each other by adhesion protein, and inside each cell there is an actomyosin network, like has been described by Eva this morning. And so the forces are generated inside this network and are transmitted from cell to cell to the scale of the epithelium. And so this process involves different scales, and we are interested in understanding how physics can allow us to connect these different scales. And there are two questions in particular that that I find interesting about this epithelia. One is the fact that in some cases they don't stay flat, they start to deform in 3 dimensions and they fold. And so we'd like to understand how the forces generated in the cell give rise to mechanics of the surface, which can deform in 3 dimensions. And the other aspect is that within the plane of the tissue, the cells move, they change their shape, they grow. And so together they give rise to massive collective flows. And that's also something we would like to understand, and eventually put these two aspects together to have physical theories which capture the deformation and flows of this tissue in 3 dimensions. So here I'm going to talk about 2 examples, one of a case of fold formation and one other case of flow. So the first thing I'm going to talk about is a project that we have worked on with Axel Behrens in the Crick Institute, and this is about pancreatic tumor formation. So this is a view of the pancreas of a mouse. So this tissue in red is the pancreas, and this is a close-up view. And what you can appreciate looking at this tissue is that the pancreas has this complicated tree structure. This is a schematic here. So as you go deeper into the tree, you encounter ducts, so tubes, which get smaller and smaller, and at the end, the tubes are capped by acinar cells, which secrete enzymes inside the tube. And the reason why this lab is interested in the pancreas is because they want to study pancreatic cancer. And they do that using a mouse model where the mouse is genetically modified so that tamoxifen injection results in the transformation of a few cells, which are distributed in the pancreas. And the transformation are such that these cells become cancerous. So in practice, they have a tumor suppressor gene which is deleted and an oncogene that is activated. And then one waits a little bit. So normally the cells of the pancreas divide extremely rarely, but these cells are more proliferative. And so after a few weeks, the cell has grown into a small tumor. So at these early stages, it's not a fully developed cancer yet. So the epithelial integrity is maintained, but there are these small tumors. And then this lab has developed a method to image in 3 dimensions the pancreas, and in red, those are the cells that have been transformed. And so with this method, one can look at the shape of the tube and look at the shape of the tumors, how they have grown inside the tube at these early stages. Okay, so what they do then is they take these images and they can do quantification of the shapes of the tumors that they have been observing. So this is a 3D view, this is a cross-section. And they see 2 different kinds of tumors. In some cases, like this one, so this yellow line is the boundary of the tube. And in some cases, like here, this red tumor has grown towards the inside of the tube. In some other cases, like here, the tube has deformed and the tumor has grown towards the outside of the tube. And interestingly, it also turns out that these tumors are probably more aggressive because they tend to recruit more cancer-associated fibroblasts. Okay, and then the question is, why are these 2 shapes emerging? And this is strongly correlated with the size of the tube. So this is a plot here showing the duct diameter, so the diameter of the tube, and whether the tumor grows extratubularly or intratubularly. And you can see that these intratubular tumors are only seen in large tubes. Okay, so the question is why? And so then talking with them, we thought that this may have to do with the distribution of actin and myosin in the cells, and here in particular, I've quantified phosphomyosin light chain. So this is a cross-section of the tissue. Those red cells are transformed cells, and these cells are wild type, and then this is a view of the phosphomyosin light chain intensity, and then one can make quantifications along the apico-basal axis of the myosin intensity. And this is, it's clear that the distribution of myosin is very different. Normally, there is more myosin apically, more phosphomyosin light chain apically, but in the transformed cells, the distribution basally and apically become more equal. And interestingly, if one grows these wild-type cells or transformed cells into organoids, the same difference is seen, so which suggests that this transformation is autonomous. Okay, so that gives us the following picture of what happens. This would be a schematic of the cross-section of a duct. The apical is towards the inside, is under high tension, if we assume that apical tension is related to how much phosphomyosin light chain there is. And then in the transformed cells, there is a modification of the distribution of basal and apical tension, and they become more equal. Okay, and the question is, what is the consequence of this? And so to do that, we have used a model that we have developed, a 3D vertex model, to represent the shape and mechanics of epithelia. And the idea of this model is to represent the tissue by a set of apical and basal vertices. So a vertex is a point that is located either apically or basally at the tricellular junctions. Then based on these vertices, we define the cells. And then when the vertices move in space, in 3 dimensions, the shape of the tissue is moving as well. Okay, and when we do this simulation, we have to say something about mechanics. And our statements about mechanics are summarized in this virtual work function. So the idea is that this is a function that allows us to say how forces generated in the cell are transferred to forces acting on the vertices. And so we have 3 contributions here. We have intracellular pressure, which we say here is constraining the cell volume. We have surface tension generated by the actomyosin cortex, and line tensions that can be generated directly at the interfaces. Okay. Okay, and then we take this model and we make a simulation of the pancreas by generating tubes of different size just by changing the number of cells. And because we also try to reproduce the aspect ratio of the cells, this constrains our ratios of tension on different interfaces on in the cells. Okay, and then in this simulation, we take one cell randomly. We say now this cell is transformed, and we modify the surface tension apically and basally, as we see in the experiment, based on the myosin light chain quantification. And then we say now this cell is proliferating and undergo a few divisions. Okay, so we have essentially no other 3 parameters, and then we just run the simulation, and we see the same thing that in experiments. In small tubes, we get these outgrowing tumors, whereas in large tubes, we get these tumors that grow inside. So to quantify this a bit more precisely, this is a plot showing the deformation of the tumor as a function of the diameter of the tube in experiment and in simulation. So these positive values indicate deformations that are outgrowing, and negative values deformations that are inside. The colors correspond to 2 different times, and in both simulation and experiment, there is a threshold of duct diameter where one switches from one deformation to the other, which is about 70 micrometer. Okay, so we thought, okay, now we can use the simulation to make a bit more tests. And in the simulation, we do 2 things. We say that the cells over-proliferate, and we say that they change their apical and basal surface tension. So we can remove one of these things and say, now let's say that the apical and basal surface tension don't change, and we just over-proliferate. And we run this simulation and quantify the deformation. And we see that in that case, we lose this transition. All the tumors, artificial tumors that we simulate, grow towards the outside. And we can test this experimentally because it turns out that if one does only a deletion of the tumor suppressor gene, the cells over-proliferate, but they don't change their myosin distribution. And then we see the same thing. We see this loss of behavior. All the tumors now grow outside. So this behavior of growing outside or inside is really dependent on the redistribution of actin and myosin. Okay, and to understand a bit more about this, we can use a continuum theory. So basically, the vertex model simulation shows that it works, but we don't know why. So it's easier to understand why, to replace this description at the cellular level by a description of a tissue as a surface and using a continuum theory, where one can recognize what is going on more easily. Okay, and for that, we can use the theory of active surfaces that we have developed with Frank Julicher. And the idea is to describe a surface where you have a cross-section of the surface, you have some stresses, they sum up into a tension, but also differential stresses acting along the cross-section of the surface result in a torque. And this torque can be passive or active. And in particular, in an epithelium, the difference of apical and basal surface tension creates an internal active torque that depends on the difference between apical and basal tension. Okay, so in the cross-section, there is a tendency of these forces to make the cross-section rotate, and it makes sense that it's dependent on this difference. So now we can do another very simplified model of the tumor growth, where we have a cylinder, and now the fact that there is a tumor is represented by this red region, which is going to grow, and also is under a difference of active torques because there is a change in the difference between apical and basal surface tension in this region and in this region. And then if we do this calculation, we see the same thing, that according to the radius, the direction taken by the tumor changes at a critical radius, and above this, or threshold radius, above this threshold radius, the tumors grow inside, and below they grow outside. Okay, and the threshold radius depends on the difference of active torques, such that if there is no difference of active torques, there is no transition. Okay, and looking at this equation, we also see that the threshold comes from the competition between cell growth and active torque. And so what I mean by this is there are two effects. One, which is that because of cell proliferation, locally the cells increase the area, so they locally increase the radius of the tube, so that favors the growth towards the outside. But at the same time, because they change their apical and basal surface tension, they acquire a spontaneous curvature, which tends to bring them towards the inside. So this effect here tends to make the tumor grow inside, and it occurs more easily in a large tube because the resistance to deformation is less, and so physically that favors the growth of the tumor towards the inside. Okay, so that was about folds, and the other thing I wanted to tell you about is ongoing work that we're doing with Nick Tapon, also in the Crick, on a histoblast nest. And so this is about flows. And so now there is no fold formation, so well, maybe a little bit, but the shape of the tissue in 3 dimensions is not changing very much. And so this is a movie of the abdomen of a fly during the pupal stages. And so these different, this is the beginning, you can see these big cells here are larval cells, and all these small cells here are different nests that exist in different hemisegments of the fly. And what we see in this process is that the histoblast nests start to grow, and the larval cells die, and the histoblast nest finally invade completely the epithelium. And in the adult fly, the abdomen is entirely made of these histoblast cells. So it's quite a fascinating process of tissue replacement by another. Okay, so we want to analyze what's going on in these tissues, and so to do that, we make a movie where we see more clearly these two nests, and then we can segment this movie. So this is an example of segmentation. And in addition, we here have labeled the dividing cells. Okay, so, and I should say it's more challenging than usual to quantify, to segment these tissues because the cell division makes it very heterogeneous. So we have used, with Matt Smith in my group, we have used deep learning to help us doing this. Okay, and then we want to analyze these movies, and we need a way to quantify what is going on. And in collaboration with Susan Eaton and Frank Julicher, in the last years, we have developed a few methods to analyze the segmented movies of tissues using a triangulation method. So connecting the cell center by triangles, then analyzing the deformation of this triangle, and defining mathematical quantities that allow to relate the cell deformation to the tissue deformation. So here we are doing a relatively simple version of this. So we have done different things, but what I'm going to show here is a simple version, just asking how the tissue area is increasing. And there is a simple decomposition equation because the tissue area can increase only if each cell changes its area, or if the cells keep the area, but they divide. And this is captured by this decomposition equation, saying that the rate of tissue area increase is the sum of the cell area increase, the division rate, minus the delamination rate, which acts opposite to the division rate. Okay, and then we can take the movies. We have 3 different wild types, and they look similar. And we look at the rate of shear as a function of time. So this is how much the tissue deforms as a function of time. And this is the cumulative contribution, so how much the area actually increases as a function of time. Okay, so the rate is decreasing, so the growth is slowing down. And this is the overall logarithmic area increase. And some of this change comes from cell area, and some of this change comes from cell division. And in fact, the cell area is not doing much. There is a small increase at the beginning, but then it relaxes to 0. So the cell area are increasing a little bit, but then it's constant. So most of the growth is due to cell division. Okay, and if we look in more details at the, sorry, I forgot to say there is very little cell delamination in the histoblast. Okay, if we now look at the dynamics of cell division, we see that it has quite a complicated pattern. First, the cell division rate as a function of time oscillates, and then it decays. Then we thought, okay, we want to understand this, and there's been some, there's generally the idea, which makes sense, and also been shown in some tissues, that the cell division rate might be sensitive to geometry, in the sense that a small cell will divide slowly, and a large cell will divide more quickly, and this allows to maintain homeostasis, because since you have the cells at division, that allows to maintain the area of the cells, or the volume of the cells around a fixed value. And this can be captured, or is generally captured in physical models by saying, as a function of the cell area or the cell volume, there is a rate of cell division minus a rate of delamination, and there is an increasing curve such that for a large area, the cells divide more quickly. So now we have thousands of cells, so we can test this, and we see there is absolutely no correlation in this tissue. This is the cell lifetime as a function of the cell area. I mean, I'm even amazed that it's so flat. It's extremely flat, no correlation. So the cell lifetime does not depend on the geometry, and we also tried other quantities like the cell elongation, and also absolutely no correlation. So the cell division rate or the cell cycle lifetime seems to be independent of geometry. So then we said, okay, let's just look at the distribution of lifetime, and of course there is a probability distribution. It's relatively peaked. The average is about 4 hours, and there is a coefficient of variation of about 20%, so some uncertainty in this cell cycle time. And then there is based on this, there is a very simple explanation for these oscillations that we see, which is a loss of synchrony. Because if you take one cell, you let it divide, and then you let it divide again, they will be slightly offset from each other, and then you let the process continue, you lose eventually the synchrony between cells. And so, in fact, at the prepupal stages, there are a few synchronous divisions, which give rise to relatively large groups of cells, which presumably are synchronized. And then there is this process that we observe that starts where cells are slightly desynchronized. And if we simulate this, we do see this oscillation, which are the remnants of the progressive desynchronization of the oscillation. So this we can explain. But what that doesn't explain is the decay, finally, in the cell division rate. Okay, and here the surprising thing is if we quantify the cell cycle lifetime, we see that it's constant. So this is the cell division rate as a function of time, it decays. This is the cell cycle time, it's constant. So this seems contradictory. And the reason why it's contradictory is because there is not only one type of cell. This is what I'm showing now. So all the colored cells in this movie are the cells that we can follow during the entire movie. The pale green cells are a bit special. They will become SOP, so they differentiate in a certain type. And then the dark green cells are cells that will divide, and the red cells are cells that will not divide, so become quiescent. And what we can see is that all cells that divide have the same cycle lifetime on average, but there is an emerging population of cells which are quiescent. And if we compare it between different, so there is this progressive covering by these quiescent cells, and if we compare between different movies, it's not, there is no clear correlation, spatial correlation between different movies, so the apparition of these cells appears to be stochastic. So what we do then is that we have quantified the probability for each division to give rise to one quiescent cell. And so P is the probability to give rise to 1 or 2 quiescent cells, and alpha is the probability given that you create a quiescent cell that you create 2 quiescent cells. And there is this increase over time, this switch-like behavior, where the probability is initially 0, and then it increases to 1. So the question is that enough to understand the dynamics of growth? And the answer is yes, because we can do a simulation where we take one cell, we make it divide, and then as a function of time, there is a changing probability that each division gives rise to a quiescent cell. And if we simulate this process, we get a graph like this. This is the total number of cells, and this is the number of quiescent cells. And this is the apparent cell division rate. This is in simulation, this is in experiment, and it looks very similar. So this essentially captures what is going on. But then the question is, how do we get this increase of probability of become quiescent? And this we don't really know, but we have some indication. One of them is that, again, it's not sensitive to geometry. This is a plot here showing as a function of time the area of dividing cells. In blue, these dividing cells will give rise to dividing cells. In red, they will give rise to 1 or 2 quiescent cells. And the area is not different. So there's no selectivity such that the small cells would give rise preferentially to quiescent cells. And if we look at cell elongation, there's also no clear difference. So no input of geometry. But the interesting thing is then we thought, okay, is it possible that the neighborhood of a cell influences its choice? And so to do this, we do this quantification. So for each dividing cell, we measure in the neighborhood how many quiescent cells there are. So there are 0, a lot, or intermediate densities. And then we plot the probability of these cells to become quiescent depending on this density. And so the blue cells are the cells that are isolated from quiescent cells. The red cells are full of quiescent cells around them. And there is a clear gradation. So the more you are surrounded by a quiescent cell, the more you have a probability to become quiescent yourself. So it doesn't explain everything because these probabilities are also increasing. But it seems to be that some aspect of how this tissue switches to this quiescence involves communication between nearest neighbors. Okay, and so to summarize, the area is constant during most of the growth. There are oscillations that are just due to loss of synchrony. The growth stops by stochastic switching process to quiescence, and the switch appears to involve some communication between neighbors. Okay, and with this, I would like to thank people in my group. Sylvanus has done the work on the pancreatic tumor formation with the help of Diana, and John Willesum and Matthew Smith have been working on the histoblast, and thank you for your attention.